to start our afternoon of presentations. I am going to, if you're just joining us this afternoon, welcome. We've had a wonderful morning so far of speakers, and I should have thanked all of our morning speakers before we left, but I forgot. But thanks to all of our morning speakers. If you're still with us, phenomenal talks, and I expect nothing less from our afternoon speakers as well. So I just popped the agenda for today into the chat box. So if you want to follow along, see what's coming up, click on that link. Just a reminder as we get going today, if you have questions for our speakers or when you have questions for our speakers, please use the Q&A button at the bottom of your Zoom window in the Zoom ribbon there. And please use the chat box for, hellos to and from folks, any technical issues and things like that. But it really makes us easier to kind of shuffle through those questions if they're all in that Q&A box. All right, I think that's it. Other committee members, if I'm missing anything, pop it into the chat, but I don't want to talk any longer so we can get on with our afternoon agenda. And first up, we have Jeff Brown. So Jeff, I mentioned this earlier in the morning. He is our most recent addition to our Ohio Bat Working Group Planning Committee. And it's been great to have him as one of the team. He's a program manager with Environmental Consulting and Technology, and he has very graciously stepped up to help organize the 2022 Bat Blitz. So he wanted some time on the agenda to just kind of share a little bit more about that plan and what we will hopefully have in store later this year. So thanks, Jeff, and I'll turn it over to you.
All right, thank you, Marnie. And thank you everybody for being here virtually. We wish we could have done this in person. Makes the question and answer during lunch and everything a little more fun, but this is very good as well. So I wanted to give an update on the Ohio Bat Blitz. We are moving forward this year. Let's see if I can advance a slide. So just wanted to give a little bit of a background. So initially back in 2019, we had some discussions with other organizers of Bat Blitzes, primarily in Kentucky, some in Tennessee. They gave some helps on here are some big items that you need to deal with. You need to find a good location where that would support a lot of, multiple misnet locations where people aren't too far from each other, that the data would be useful, maybe where you even have data gaps. You need to pick a good date that you can get a good turnout. You need to think about funding, how you can support folks that would be traveling in and any of the needs during the Blitz itself. And as well with that is tied is lodging, where folks would stay, what amenities would be close by, and those types of items. So the Ohio Bat Conservation Plan Research Committee, I think we discussed these and looked at various records, came up with some locations, about 6 of them with data gaps, large enough to support a Bat Blitz. And that was in September of 2019. And then we engaged Ohio Bat Working Group Research Committee in February 2020. Then COVID hit, everything went on hold. This picked it back up really in December 2021, just a month ago, and now we're on for 2022. We feel that we can do it safely with people. We can do it safely for the bats as well. The folks who were on that initial February 2020 committee meeting, having the discussion, I put their names over there. So it's a good diversity of backgrounds from whether or not they're state or federal or consultants or a park district. And we really appreciate everyone's input in the decision-making going into this. But again, we're on for 2022, and our location is going to be Germantown Metro Park. We're really excited. We've been working with Grace Deitch and her folks to really identify some locations where we might be able to net there. This is a metro park that's in the system for Five Rivers Metro Parks, which is really around Dayton, and surrounding maybe more than just Montgomery County, but it's a really nice park system. And if you go to metroparks.org, there at the bottom, you can click on Germantown. You can kind of see a little bit more of the details of this park. It's in Montgomery County. It's about 20 minutes west of Dayton. It's about 3,500 acres and it's bisected by the Twin Creek. What's nice about this location is the Twin Creek watershed is the same watershed that the Preble mine is located in. So as Joe mentioned earlier in his presentation, we haven't been in that mine to census for Indiana bats or Lucy's or anything else in quite some time due to just some access issues. And so we don't know what that looks like. It used to have upwards of 25,000 bats that were using that, thousands of little browns, hundreds of tricoloreds. At the height, it was maybe 10,000 Indiana bats. So it's within the watershed. It's a great habitat, and hopefully we'll be able to capture some of these as maybe they're moving towards that location to hibernate. This is just a general location map. I know it's out of context. North is straight up. Germantown, the town itself is down to the south and east. As you can see, it's a pretty large contiguous forest type here. And we've identified a few of these locations here with the push pins where we think that mistnet sites would do well. We've got a couple that are on the Twin Creek itself. The challenge with the Twin Creek is really accessibility. We'll have to pay attention to what the weather's like, if the river's up or not. I guess 2 years ago, we did set detectors out for about a month in August, kind of where you see the dots, the bullseyes. And we had a lot of success capturing information at these locations. We had a total of about 1,200 call passes by Lucy's, 225 total of Sedalis, and about 300 or so of tricolors. We didn't have very many septentrionalis at all, only 11. So this wasn't manually vetted. It was just kind of run through Kaleidoscope. And so I'm just kind of regurgitating that information back to you without a lot of detail in it. But with some of those numbers, certainly we have a strong chance of capturing Myotis species. And then, of course, big browns were, 8,000. Red bats were 1,800 in the call passes that we did record. And so the date that we're going to do this is August 19th and 20th of this year. So I put the calendar on there so you can all see it. The 15th is kind of the day that is kind of the deciding factor on this one. It's at the end of the maternity season for folks to net, whether maybe it's for research or for projects dealing with Section 7 surveys, presence, absence. It's also a weekend before some of the universities go back to school and start their classes. So it just seemed to be a good weekend. We may compete with other states, I'm not sure, but I'm hoping that we can still draw a pretty good crowd and gather some good data here. It provides for the most participation by the most people. Funding, sponsorship is needed. We're going to need some help with this. The goal is to provide lodging for at least site leads. We want to provide white nose syndrome decon materials and some limited snacks, drinks, water during netting, and maybe after netting at the appropriate place, some festive adult beverages. Five Rivers Metro Parks is the host, and they're providing a generous sponsorship that's going to help, but we still need additional funding to really make this all that it can be. And some of that can be in money and a donation of money so we can get the lodging for folks. It can be in donation of maybe at the end of the year, white nose decon materials, nitrile gloves, Clorox wipes, baggies for handling bats. It could also be transmitters if folks have extra transmitters and they're not going to use them this year. And those are things that we can use. And more to come regarding how to sponsor and what maybe we may ask for. We're going to be developing that here in the next couple weeks, get that to folks so we can really start moving forward with some of the details of that. So please consider being a sponsor. That would be greatly helpful. And it's through the generosity of event sponsors that we'd be able to provide some of the lodging. So again, I'm going through the big high topic items that were kind of presented. We're looking at lodging near the Dayton Mall area. It's Miamisburg, West Carrollton. It's about 30 minutes, about 14 miles to the east of Germantown. It's located between I-75 and I-65. It's right there on the interstates. So folks either coming in or when they're leaving, they'll be able to jump right on the highway and get to, their destination from there. We're going to, work to secure a block of rooms and get a decent price. And again, a sign up will come out again with a little bit more detail of who may be interested and available to participate. And my contact information will be coming up here, but please feel free to email me at any time, any of the questions. There's numerous amenities near the Dayton Mall area. There was some discussion that Germantown does have some camping available. It might be an opportunity for some folks that want to camp. In mid to late August, who knows what the temperature is going to be this year? Could be hot as all get out. It could be nice camping weather. We won't know until we get there, I guess. But again, please let us know if you're interested in being a sponsor. And just some other information, please consider helping with planning. So myself and Sarah, we're kind of tag teaming this thing and trying to move it forward. I anticipate that we're going to need help from other folks. We need to deal with issues regarding safety not only for our volunteers, which Grace and her team is going to help with, but also for the bats as well. It would be great, if we can set up a decon location and have volunteers ready to just help decon, so the folks who have been working the nets for the evenings can kind of just relax and let that be addressed. We also have different permits that we're going to need to get. We're going to have to work through some of the issues with funding and lodging. The goal is to provide valuable data for our host, Five Rivers Metro Parks, as well as Fish and Wildlife Service, Ohio Field Office, and ODNR, Division of Wildlife. So this is information we can fill in a gap. Again, we're on that watershed with the Preble Mine. It could be good information to help us understand if the bats are still even in the watershed in good numbers. It provides a learning opportunity for students and the public regarding bats and their important role. We want to be able to put some transmitters on some bats so we can track. That's a great opportunity to teach people how tracking bats to roost is done. We can see maybe where they're moving in the evening. We might see that they're moving on from the whole area within a night. But it's a great learning opportunity. And I know that the Metro Parks folks really want to get the public involved and have one of the locations accessible to the public, so folks can register and come and see what's going on. It's an important part of their mission, and we want to support that the best that we can. And ultimately, it's building a closer bat community in Ohio. So I got this big long run-on sentence here, but I really do feel that, you know, through communication and collaboration between bat-minded people, we keep common bats common and do our help to recover federal and state listed species. So I've rambled there, and I think that probably takes me to my time. But again, email Sarah and myself if you have interest in joining. I'll answer any questions that pop up in the chat after I'm done here, but I hope everyone will consider participating. All right, thank you very much.
Thanks so much, Jeff. And if you wouldn't mind putting your email and contact information in the chat box, just so it's easily accessible to everyone again. Please, everyone, if you're interested in helping, reach out to Jeff and Sarah. All right, let's move on to our next presentation. Next on the agenda, we have Nathaniel Marshall, who is with Stantec Consulting. And he is going to be presenting work that he and James Kaiser, also with Stantec, have done using environmental DNA from water-filled road ruts to detect bat occurrence. So I, for one, am very interested in hearing more about this project and how it was done and what they found out. So thanks for being here, Nathaniel, and I know James is here too. Nathaniel, I'll turn it over to you. Thank you so much.
Yeah, thanks for the introduction. Here, let me share my screen. Can you see that?
Yes.
Perfect. All right, so I am Nate Marshall, and yeah, I'll be talking about the detection of bats with environmental DNA from road ruts in upland forest. And this work was done with Stantec, with my two co-authors here, Dan Simons and James Kaiser, who are both with Stantec. So the main purpose of this study was to determine if we can, in fact, detect bats that are potentially drinking out of road ruts in upland forests where it's particularly perhaps far from other sources of drinking water. So we know that it's more likely that they would be targeting these road ruts for a drinking source. So just to give a little bit of background here, environmental DNA is really a mixture of genomic material that all organisms are constantly releasing into their environment. And this can be a combination of urine, waste, mucus, hair cells, a lot of different skin cells, that type of stuff. And you can really use a lot of different sources for your eDNA collection. There's studies with soil, sediment, water. There's recent studies coming out for detecting eDNA that's floating around in air. And particularly with bats, there's a lot of research with guano. And that perhaps can fall under the definition of eDNA, depending on how you want to define it and who you're talking to. But in our sense, we're not really defining a guano sample as eDNA because that came directly from the bat, and you have direct evidence that there are bats in that location. So really, we're talking about a collection of a sample where the DNA, you don't know you're directly collecting the DNA. And for really the inspiration for this study is there's been a lot of work over the recent few years where different terrestrial animals are being detected from water eDNA, where they are known to either enter the water or to take drinks of water. So we have different mammals of recently, and then even a bird, this finch species, was detected from areas where they were known to be at and drinking out of the water. So this really inspired my co-author James Kaiser to think about how this could potentially work with bats, because through his bat surveying work, he's noticed that in a lot of areas, bats rely on road ruts for drinking water. So if that's the case, we have these small puddles that we know bats are using. Perhaps there's a chance that their eDNA is entering those small puddles, and we can detect them. And another thing he's noticed is that not only are common bats using road ruts, but a lot of rare species use road ruts. And so because of that, it might be a nice source of detection for rare species that are sometimes hard to detect. So here's an example of a bat coming down to take a drink out of a water source. And you can see they sort of skim their tummies or their bellies up against the water. And through that process, they may be releasing hair, saliva, or skin as they're taking a drink. And then perhaps even in some occasions, some urine or feces may enter the source of drinking water. And all these different types of material or cellular material will have genetic, genetic information in them that we can link back to the bat that the DNA came from. So there's quite a few benefits eDNA might provide in terms of sampling for bats. When you're setting up these traditional mist nets, you have some potential risks with that to both the survey staff and the bats themselves when they're being caught in the nets and being handled. And of course, you need potentially, in some of these occasions, multiple crew members to set up these mist nets. And then you need time to wait for the bats to be caught. So if you can go out with just 1 or 2 people and collect water samples, that's potentially a time and cost saver. And then acoustic sampling is also a non-invasive form of sampling, just as eDNA would be. But sometimes acoustic sampling can miss species depending on the acoustic database. Some bats have multiple types of calls, so they can be missed or misinterpreted for another species. So potentially eDNA can just add additional information on what bats are in the area. So if it's another survey tool that helps our understanding of bats, I mean, that's great. It can be an additional complementary tool. So for this project, we sort of had 2 pilot studies, and these were broken up into Kentucky and Ohio. Our first study occurred at 5 sites in Kentucky in late May, and we sort of were just wondering, hey, can we detect bats from these road ruts? And then with the EPRI and AEP, we were sampling 15 sites in Ohio in late July, excuse me, on the recreation lands in about central Ohio. And then we went back to 2 sites in Kentucky and resampled in late August and compared that to the findings in May. So here's a map of the sample sites in Kentucky. This was in southeastern Kentucky. And we had 5 sites that were across a couple different counties. And what you can see here is that the purple sites were sampled in only late May, and the blue sites were sampled in late May and again in late August. So here's an example of a road rut here filled with water. And yeah, we had 5 sampling sites across 3 different counties, McCreary, Bell, and Harlan County. And again, we sampled in May. And the question was, can we detect bats from these locations where we know there's a decent density of bats? We also did a control experiment to sort of make sure that eDNA was slowing off of bats while they were drinking. So we did a simulation of bats drinking in this Tupperware tub, where we took 3 different species of bats, big brown bat, eastern red bat, and eastern small-footed bat, and simulated 5 drinks. So to do that, just sort of skim the belly of the bat across the water. And we did this 5 times for each species. And then we filtered that water on 2 different filter types to see if there was a difference in detection across filters. So there's a picture of the bat simulation. And this is a map of the Ohio sampling sites, where the blue triangles are the 15 eDNA sampling sites. And then at some of these locations, we also had acoustic data, which is somewhat of an interesting comparison to see where there was a high density of bats compared to eDNA. And here's another example of a road rut filled with water across the recreation lands in Ohio. Again, 15 sites sampled in late July. And one thing to note with this example here, this is a road rut that was out in the sun for a while some of the other road ruts are smaller in size or maybe deeper in size and have vegetation completely around them. So there's a variety of types of road ruts that we sampled. For the eDNA analysis, we used 2 different approaches to try to detect bats. The first approach is called a quantitative PCR or a qPCR. And for that approach, it's you're actively trying to target a single species. So you only are interested in one particular bat species. The other approach we use is called a DNA metabarcoding approach. And this is really a community approach. It's sort of a passive sampling of eDNA. You're just trying to get an idea of all the vertebrates that might be present in your sample. In previous eDNA studies, it's been shown that qPCR tends to have higher sensitivity for species that are rare. And that's just because you're sort of actively targeting that species. Within the DNA metabarcoding, we actually use 2 different primer sets. So these are 2 different sort of genetic assays to try to detect bats. One we used from the Walker Lab at Northern Arizona. This primer set was developed for analysis of bat guano. And then another one we used was developed for eDNA studies for detecting aquatic vertebrates in rivers, but has been shown to work well with terrestrial vertebrates in many cases. For qPCR, we used 2 different qPCR assays, one that targeted big brown bat, as we knew there was a high density of big brown bat in some of these areas. And then we also used one that targets eastern red bat, but keep in mind that this actually also works on silver-haired bat. So we can't necessarily distinguish a detection for eastern red bat from a detection for silver-haired bat. But we knew eastern red bat was in the locations where we were collecting water. All right, I'm going to jump into the results now. So for the control bat dip, I'm going to start with the DNA metabarcoding approach. So this is detecting all the vertebrate DNA in our sample. We collected 4 total samples. 3 of them successfully worked in the lab. And all 3 of these samples detected big brown bat and eastern red bat, while only one of them detected small-footed bat. And then the other thing to note here is that you can see this is sort of broken up as the percentage of DNA within each of these samples. And we can see that big brown bat was majority of the DNA for all 3 samples. And we used 2 individuals of big brown bat versus 1 individual of the other 2 species. So it makes sense that there was more big brown bat in this control sample. It's a little interesting that only 1 sample detected eastern small-footed bat. I'm not quite sure why that was the case. Now, looking at the qPCR, we used 2 different assays for qPCR. So the first one was looking for just big brown bat DNA, and we did detect big brown bat DNA from both filter types. So this is showing the 2 micron filter and the 11 micron filter. So this is a plot showing CQ value, which is the cycle quantification. So it's basically the point at which you're detecting DNA in your sample. And it's actually the opposite of what you would think, in terms that a lower CQ value means greater concentration of DNA. That's because you're detecting the DNA faster. So the 11 micron here actually has more DNA than the 2 micron filter size. So both filters work to detect big brown bat, but we found higher amounts of DNA on the 11 micron filter size. For eastern red bat, again, both filters worked, but similar to big brown bat, we found greater concentrations of DNA were found on the 11 micron filter. So it appears that the 11 micron filter was collecting more DNA compared to the 2 micron, which you might think, well, that's weird, it's a larger size. But eDNA, particularly in this case, was probably a lot of hair falling off the bats. So that's definitely going to collect on 11 micron. That's hair's not going to be small enough to fit through that. So what we're seeing here is probably a difference in the filter type, which were a polycarbonate matrix versus a glass microfiber matrix. And what it came down to is probably some sort of chemistry relationship during the lab processing, not necessarily the filter size. So doing some more investigation of this with using the same type of filter would be an interesting comparison. We can also see that for either filter type, big brown bats displayed greater concentration of DNA compared to eastern red bat, which is what we found in the metabarcoding data set as well. So that makes sense because, again, we used more big brown bat individuals. We would expect more big brown bat DNA in the sample. All right, now I'm going to get into the environmental samples. So here we're talking about the actual road rut samples. Using the DNA metabarcoding approach, so this is the community approach where we're trying to detect any vertebrates, we did not detect any bats in these samples. From Kentucky or from Ohio, we unfortunately just had no bat detections. But we detected a number of other vertebrates. We detected some amphibians, birds, and mammals. So a number of toads, frogs, even a newt, an eastern phoebe, and then things you would expect such as possum, deer, raccoon. So a number of expected vertebrates in the area, but it shows that these road ruts have animals using them for a number of reasons. They'll be leaving behind DNA, and they can be used as a nice biodiversity assessment. Now for qPCR, this is more sensitive typically than metabarcoding. We did detect big brown bat and eastern red bat in these samples, or in a few of these samples. For big brown bat, we detected this species in one sample from Kentucky collected in late May, and that same sample again collected in late August. So we were very confident that this species, was in the area. It was detected across 2 different months. And then for eastern red bat, we detected this species in one sample from Kentucky and then one sample collected in Ohio. Now, the sample in Ohio showed very low amounts of DNA, but what's interesting is that it corresponded to this site that had the highest acoustic data. So our only detection in Ohio came from the site that had, from acoustic data, suggested the highest density of bats. So this is exciting that we're getting detections, but also brings about the question of, well, how many bats are required to be in an area before you detect them? And while we know rare bats are using the road ruts, we also know that their populations have decreased since, this figure over here since 2000. So if the bats we're looking for aren't in the density needed, will eDNA even provide a sampling opportunity or a sampling option? So really quick, I'll just end with some conclusions. The control bat dips detected all 3 species, although eastern small-footed bat was only in one filter for some reason. We didn't detect bats with a community approach, but we did detect a number of other vertebrates, which may be useful in certain circumstances. And qBCR, as other studies have shown, was more sensitive in this case, and you can design new assays that target any species you're interested in, such as any threatened and endangered bat species. So we had some success for 2 species. We'd like to expand upon this for other species that we know would be likely to use road ruts. We can really optimize these sampling practices. I think increasing the number of biological replicates collected from a road rut will increase your likelihood to collect eDNA of a bat, and then thus increase your probability of detection. I think it'd be interesting to sample across a density gradient so that we know, okay, after this density of bats, we no longer confidently detect them with eDNA, and we can better understand the feasibility of eDNA for this purpose. And then, as I showed, there can be a lot of differences in the environment around a road rut, and all of those will impact the longevity of eDNA and thus your ability to detect it. So sampling around different road ruts and understanding those characteristics might improve future surveys. And then I'd just like to thank a lot of different partners through this work. We had funding through AEP and through EPRI, which were great partners for developing the plan here. And then sampling help through the Kentucky Department of Fish and Wildlife, and our genetic lab partners through Northern Arizona and Genadax to analyze the eDNA samples. With that, I'll take any questions if I have time.
Thanks, Nate. Very, very interesting stuff. And there is a suggestion by Jeff as to whether or not we can incorporate this into the Bat Blitz. So food for thought.
Yeah.
We are awesome. We are about 5 minutes behind schedule, which is fine. We've worked in a little bit of a buffer here later on, but that does mean we have time for question. And Melissa Moser is going to kind of facilitate our Q&A for this afternoon. So I don't know if we have any, but Melissa, I'll turn it over to you.
Yeah, I actually don't see any questions. There's a little bit of chatting going on about the wilds collecting DNA samples, but you know, in mentioning the Bat Blitz, potentially collecting eDNA there, but otherwise no questions right now.
Hey, Marnie, this is James Kaiser. I
Hi,
would
James.
like to also thank Dr. Joe Johnson's lab at Ohio University. He provided some samples from red bats and silver-haired bats for us to develop the primer for the QPCR. So,
Wonderful.
and I'd just like to say that the technology, I think, is up and coming, but we probably missed out on a lot of great opportunities when we had 70% of the captures over road ruts for northern long-eared bats. And so I think this tool may be very useful when we start looking at trying to find northern long-eared bats back out on the forested landscape. It might make it a cheap, good way to do that.
Awesome. Thanks, James. All right. We are going to move on to our lightning talk session. So these are going to be fast-paced talks. We have 4 speakers, and each of their presentations are going to be 4 minutes long. We are going to wait to hold questions until the end of the entire session, and then we'll have about 10 minutes or so. So I guess if you're submitting questions in the Q&A, if you could possibly put the name of the speaker before the question, just to make it a little bit easier on us, though I think for the most part, we'll probably be able to figure it out. And then speakers, I will introduce each of you. And then if you can kind of see the video pane, when you see my picture pop up, that means you have about a minute left. So just to kind of give you that little timing right there. Okay, so first up, we have Rachel Handy. Rachel's with Oregon State University. She's going to talk about the importance of collaborating with rehab centers to enhance bat care, research, and conservation. So hello, Rachel, and thanks for being here.
All right. Hello, everyone. Is my sound coming across all right?
You sound great.
Okay, perfect. All right, let me get this up here. From beginning. There we go. All right, well, good afternoon, everyone. My name is Rachel Handy, and as Marnie mentioned, I'm really excited to share with you the potential opportunities that collaboration with wildlife rehabilitation centers can offer us in order to enhance bat care, research, and conservation. So I'll give you a little bit of background and a summary of my research. So we all know that many species of bats are facing a variety of threats for many different reasons, from white nose syndrome to wind turbines. But what many of you may not realize is that there is a really unique opportunity to learn more about these challenges by teaming up with perhaps a surprising partner, the Wildlife Rehab Center. So bats are frequent patients at wildlife rehabilitation centers, and because of that, they offer many opportunities to advance the knowledge of bat biology via collaboration. So a little background, what exactly is wildlife rehabilitation? Generally, it's broken down into 3 phases: rescue, rehabilitation itself, and of course, the ultimate goal to release those wildlife critters back into their natural habitats in the wild. So wildlife rehab centers are located across the globe in all different countries, and including in all 50 states here in the US. Within Ohio and the surrounding states, you can actually find 167 permitted wildlife rehab centers. So how do bats fit in? Well, in some cases, the number of bats entering these rehab facilities is quite staggering and is also on the rise. For example, at the Ohio Wildlife Center, located in Columbus, over a 15-year period, 777 bats across 7 different species were admitted, of which 33% were rehabbed and released. Some more recent numbers: in 2019, 220 bats were admitted, which was a whopping 95% increase from the previous year. In 2020, numbers went down a little bit. There were 141 bats admitted, but 35% of them were released. So with the number of bats entering rehab centers climbing in many cases, these facilities are really well positioned to contribute to bat conservation, especially since proactive approaches to conservation are more effective than reactive strategies. So some examples of research opportunities include data collection, more on that in just a second, actual live bats in hand, and of course, biological materials. Rehab centers have definitely no lack of materials: guano, fur, all kinds of different samples. So what's interesting is that almost all rehab centers are required to keep track of patient and presenter records in order to maintain their permitting. So one example database I'd like to highlight is Wild One. This is a free system that was created by the Wildlife Center of Virginia. And Wild One currently has at least 107 actively participating organizations across 5 countries. That's a lot of information being collected, which has the potential to reveal all sorts of different emerging threats and trends. And here are just a couple examples of some of the recent stats from the database in general regarding bats. So you can see there's a lot of data really to be found. And furthermore, this data has not really been well mined yet. So for those wishing to collaborate with rehab centers, it offers these researchers really a chance to gather a ton of information and hit the ground running. So collaboration between rehab centers and potential partners could enhance our knowledge of bat biology. Furthermore, because many of the conservation concerns threatening bats are common across many different species, collaborations on studies of bats can have far-reaching effects. So some brief examples here could be scientific researchers, like I mentioned before, the information we can give to them, educational partners, schools, university students, governing agencies and policymakers. This may include environmental consulting firms, could be grouped in there, and of course the community at large. So briefly, I'm going to go over with you some potential research questions that could come up or that some researchers have thought of. So for example, what are the most common injuries and sources of mortality for bats entering these facilities, and how do they vary across landscapes? Another one might be what pesticides or other toxins are present in bats, and do they vary by foraging strategy or maybe rescue location? And one that I'm really personally interested in, what is the post-release survival rate of rehabbed bats over the short and long term? And do these rates vary by species, injury type, and geography? So this was just a very brief overview. So I'd like to thank my supporters, Oregon State, the Ohio Wildlife Center, the Wildlife Center of Virginia. I'd be happy to share my full presentation. You can reach out to me with any questions or comments. I have my information there. And thank you all so much. Have a great day.
Thank you, Rachel. That was awesome. Okay, next up we have Lucy Williams. She's with Ohio University, and she's going to be sharing some of her work on the importance of acoustic surveys when conducting winter surveys for bats. So welcome, Lucy.
Hi, yeah, thank you. Can you hear me okay?
Yes.
Okay, great. All right, let's get this started. Okay, so yes, thanks, Marnie, for the introduction. And again, my name is Lucy Williams, and I work with Ohio University. So to start off, I'll be going over a couple of things Joe already has touched on if you were here earlier today. So over the past few winters, a surprising number of bat species have been discovered hibernating in rock crevices in both caves and rock shelters in Ohio. And just some of the species documented include the big brown bat, the eastern small-footed bat, silver-haired bats, and the federally threatened northern Myotis. And some of these species, like the silver-haired bat and eastern small-footed bat, were not previously known to hibernate in Ohio. So it's really cool that we've discovered them hibernating in the state. So visual surveys are great. We're finding these bats, but just how much are we missing with visual surveys alone? So as you can see, there are several cracks and crevices that I could not easily access without ropes or some other form of assistance to view the inside of these cracks. So there's just, and this is the same case in caves and mines as well. There are just some areas that we can't get to and can't get our eyes on. And because of this, only a portion of caves and above ground habitat can be fully surveyed visually. Therefore, visual surveys alone may be underrepresenting bat communities that are using those habitats. So in order to determine if visual surveys underrepresent bat species, and in order to compare winter activity rates among bat species, I deployed 21 acoustic detectors at non-traditional hibernacula or above ground habitat. shown in pink on this map. And I also deployed 24 detectors at traditional hibernacula or below ground habitat like caves and mines. And those are shown with the orange dots on my map here. And as you can see, I did this across Ohio and Pennsylvania. So first, I'd like to briefly talk about the species diversity we detected using acoustic surveys compared to visual surveys. Then I'll talk about our winter activity modeling. So we use paired t-tests to compare the number of species detected with visual surveys and acoustic surveys at both traditional and non-traditional hibernacula. And when comparing visual surveys to acoustic surveys outside of below ground sites alone, there is some evidence that acoustic surveys are documenting more species than visual surveys at those same sites. And this is especially true when surveying above ground habitats. We found extremely strong evidence that acoustic surveys are producing more species than visual surveys at above ground habitats in particular. And these are the same sites using a paired t-test. So I'm still hard at work collecting our data, but next I'll be talking a little bit about activity models for a couple species based on our first winter's data. So our top model for one of our more active species, the big brown bat, contained 2 fixed effects, whether bats were outside of traditional or non-traditional hibernacula, average winter temperature, which is shown on the x-axis, and we had site as a random variable. So from this graph, you can see that there is a significant effect of temperature with rapid increases outside of both habitats, but overall more activity outside of non-traditional hibernacula, which is shown in red there, compared to traditional hibernacula caves and mines shown in blue. And to compare, we have a slightly less active group in winter, the Myotis, on the right now. So for the Myotis species, you can see there's actually little difference between predicted activity outside of traditional and non-traditional hibernacula. There's a lot of overlap you can see between the blue and the red for their graph. And we found this pretty interesting because Myotis are more frequently documented using traditional or below ground hibernacula in the wintertime. And additionally, activity for Myotis species picks up after about 10 degrees and even more so after 20 degrees Celsius compared to our big brown bats, which will be active at much colder temperatures, as low as 0 degrees Celsius and even lower than that, all the way down to negative 18 degrees Celsius, which is pretty amazing. So in sum, we found that at both caves and non-traditional hibernacula, visual surveys underrepresented species diversity. Again, visual surveys are still incredibly useful and fun. And acoustic surveys can't tell you how many bats we have in an area like this rock shelter, but they can give us a more accurate representation of the species that might be present there. Acoustic surveys can also be especially helpful when documenting the winter presence of cryptic species, such as the silver-haired bat and Myotis species. Myotis in particular can be difficult to detect acoustically during the winter time. So our models could help us predict when they would be most likely detected during an acoustic winter survey. And lastly, as one of you, or as y'all may guess, we did find that activity rates will vary with species and weather conditions, of course, and we'll also be including landscape variables in our analyses in the future as well. So I still have a lot of work to do, but I just like to thank everyone that's helped this project. And yeah, thank you all for listening.
Thank you so much, Lucy. Really good information and loved all those pictures. Beautiful.
Thanks.
Okay, yeah. Again, just a reminder, if you have questions for our speakers, pop them in the Q&A and we'll address them afterwards. Our next speaker was supposed to be Mattea Lewis, but she unfortunately had something come up and she sends her regrets for not being here. But she roped Joe Johnson into presenting for her. And so he's going to tell us a little bit about the spring migration habits of little browns and tricoloreds in Ohio. Thanks, Joe. And Joe, we can't hear you. Doesn't say that you're muted. I'm not sure what's going on there. No, still can't hear you. Okay, I think Joe's going to sign out and then try signing back in. So we'll give him, hang tight for just a minute, guys. We'll see if that works for him. If not, we'll move on to the next speaker. Okay, Joe's back on. Let me upgrade you to co-host so you can share your screen. Okay.
Any better?
Yes, we hear you now.
All right, I'm gonna try and make this a super lightning talk now.
Oh,
Yeah,
you're fine. You're fine.
Totally my fault. I plugged in my additional monitor and that breaks my microphone. I don't know why. Okay, here we go. Mattea sends her regards. She did all of this work. I'm only gonna try and take some credit for it. The goal of what we did here is less a science product than it is trying to enhance our conservation efforts statewide. As you probably remember from my presentation earlier, Lucy's presentation now, and Levi's presentation last year, we've learned a lot about bats during the winter, where they are, but we know less actually at this point about where they go in summer, especially our tricoloreds and little browns. And of course, summer is where reproduction occurs and populations can rebound. So our hope is that we can track these individuals. That's what we're trying to do here, some spring migration tracking from those hibernacula to the summer maternity grounds so that we can protect and study bats in those environments. So just a shout out for how difficult this was for Mattea et al. This is a photo, you can see Levi and Lucy there as well. When we're trying to capture or harp trap during spring migration at these tunnels, we usually have a very small opening at the tunnels that are used by bats. So Mattea constructed what she calls her tater trap, her trap that makes a tunnel, exiting the tunnel out of some tarps here. And the bats will funnel through, hit the harp trap, and then we can tag them and track them during their spring migration. We tried to do this on a handful of nights in early April. Unfortunately, although we went out with high expectations and 10 radio transmitters and Joe Barber in the sky, you know, bats don't always cooperate. And instead of catching a female that you can track to a maternity roost, you might catch no bats that night, or you might catch a bunch of males. And so we were fortunate enough to catch only one female on one of the nights, one female little brown bat, on one of the nights where we had Joe Barber overhead. So our methodology here was to have Joe, the DNR pilot, if you don't know him, at a nearby airport located to our hibernacula. The moment Mattea would catch a bat of the right species, what she would do is she would send Joe a text message and say, hey, Joe, you can take off. I'm gonna start tagging this bat. In a couple of minutes, Joe will be overhead. Mattea will release the bat while Joe is over in the sky. And then Joe will track the bat while it migrates to wherever it goes, communicating to Mattea and crew on the ground so that they can try and find the summer maternity colony. And for our little brown bat that we captured the night that Joe was overhead, what it did is it went to the Clendenning Marina. And the bats here, we have a large colony that would exit and roost underneath the wooden siding and up in some space that I can't actually physically ever find them in this marina. And this was a really valuable finding because the marina is going to cooperate with us and the DNR to better preserve some roosting habitat for these bats in the long term and to set up a population dynamics study. So this is one of the values of finding these roosts, is we can try and avoid any future wildlife conflicts, and we can set up some cool research. So some more details on that one bat Mattea captured. She captured it on the 6th of April. and it ended up at the marina the following morning. It kind of took a relatively calm and collected flight of about 17.3 kilometers straight line distance to get from point A to point B. Did it all in the course of a night. And we know that this roost is an important little brown maternity site because we counted as much as 129 bats emerging on one evening. And here's just a photo of one of the little guys coming out from underneath the siding in this area. We're going to do a pit tag study in this spot, and the bats' interest in coming out all over the place is going to provide a fun challenge for us. We did catch a tricolored bat as well on a different evening. Here we didn't have Joe overhead on April 19th, but we were able to get him out in the field 4 nights later on the 23rd. And what happened is he was able to get a signal on this bat coming just under 6 kilometers from where it was tagged in the Woodbury Wildlife Management Area. Here's a look at a snag that tricolored bat roosted in for several days. And we were able to, throughout the course of the summer, determine that we have a fair number of tricolored bats thriving on the Woodbury Wildlife Management Area. And this has been an important area we've identified for future research and conservation efforts. So again, showing the benefit of working with our partners. And here they are from the Ohio Department of Transportation, who are working to preserve some of these hibernacula, Ohio Division of Wildlife for setting up the planes to track the bats and donating their time, as well as all the people who did the work, because as I said, I did none of it. This was right around the time where I had my 2 sons with my wife. And so, you know, we would just get text messages at all hours of the night. I was up anyway, so it was kind of a trip, but credit goes to all of these people. So sorry that was long and late, but thank you for listening.
That was right on time. All right, thanks, Joe. Yeah, good job. Well done. Okay, our last lightning presentation is Gregg Janos. He's with the Copperhead Environmental Consulting, and he's going to share with us a summary from over 10 years of acoustic data from driving Transects in Ohio. You sure you can do that in 5 minutes, Gregg? Challenge it's a lot on you. All right, welcome. Thanks for being here.
Thanks. All right. All right, got it. The
Looks
appropriate
good.
screen. All right. So yeah, this is a project that some of you guys have probably heard about in the past. I think Sarah's talked about it about every year in the past. But this is a project that ODNR has been working on since 2009. So they've been collecting these driving acoustic routes since 2009. And so they got a huge data set, and Fish and Wildlife is working on developing species status assessments for the little brown and the tricolored bat, both of which are endangered in Ohio. Fish and Wildlife is looking at them as candidates to be added to the endangered species list. And so Ohio's got this huge data set. They were interested in looking at this data to see what we might be able to pull from that. So here's a map of the distribution of all the routes. I believe there's 52 at the moment, so pretty good coverage across the state of Ohio. And for analysis, what we did, wrong direction, we split the state up into the ODNR districts. That way we can analyze the data kind of on a regional basis, because as we know, the habitat down in the southern part of the state is very different than the habitat, say, in the northern part. So we split them up into these districts for analysis. We processed all this acoustic data through Kaleidoscope Pro, which automatically identifies the calls for us. We then went back through all those files that were identified as a bat. Myself and another coworker went through all of those bat files and then also manually vetted them to species when possible. So after all was said and done, we had processed over 217,000 files. Of those, over 44,000 contained bat calls, and we were able to identify over 40,000 of those 2 species. And some of those files had more than 1 species that we were able to identify. So that's why our total at the bottom is a little bit larger. But as you can see, our big browns and eastern red bats were the overwhelming majority of our data, followed by hoary bats. So between those 3 species, it made up 90% of our data set. Unfortunately, not a whole lot of data for the Myotis species, our little browns, long-eareds, and Indiana bats. And we had a decent amount of data from tricoloreds. So this graph is what we're calling our call encounter rate. It is simply the amount of bat calls divided by how often a route was driven. And it's kind of a bit of a roller coaster, and unfortunately has declined a bit the past couple years, mainly the last 2, I guess. 2021, not as many driving routes were conducted as in the past. And a lot of those routes from last year in 2021 were from the districts of 2 and 5, I believe. So areas that are more ag-related. So it kind of makes sense that a bit of the call data is down a bit. This is a graph just showing the species that were recorded per year for each one of those districts. Again, fairly stable until, again, last year, but last year's maybe a bit of anomaly, not getting quite as well of a full statewide survey done with these driving routes. Since Little Browns and Tricolors kind of prompted this project, this is just a graph showing our Little Brown number of calls essentially over the course of the data being collected. It never was very high to begin with. It's kind of remained fairly stable, which I guess is good. At least it's stable, but still not very much in the way of activity being recorded on these driving routes for Little Browns. As for tricolored bats, kind of a similar outcome. Definitely have had more bat activity for tricolored bats, but it too is definitely shown a little bit more of a decline, which don't care to see, but still picked them up. Unfortunately, we didn't have any last year for Little Browns or tricolored. Little Browns or tricoloreds, but again, most of those species have been recorded in the districts 4 and the southern part of 5, and those weren't quite as much. So lastly, our common species, big browns, reds, and hoary bats continue to dominate the landscape. I think we see that with misnetting, but now also certainly with the driving transects. The Myotis bats are not commonly recorded on these driving routes, and unfortunately the tricoloreds appear to have declined as well over time. That is that.
Thanks, Greg.
Yep.
Awesome. Okay. So Melissa, I'm going to turn it over for you for questions.
All right, thanks. So Sarah asked Joe, are you guys planning to do any spring emergence tagging this year?
Did we lose? No, there's Joe. He might have stepped out though, I'm not sure.
Is that for me?
That was for you, Joe. Yeah.
Whoops.
Are you planning to do any additional spring emergence tagging this year?
I would like to, but to be honest, I've not lined up all of the stars that need to all get in order for that to be permitted, and then like the plane is just kind of like at the good graces of the DNR, and I've not lined that up. I'd like to, but I'm doing such a bad job at parenting. I haven't lined up all those stars yet. So, you know, Erin, if you're still here and you're listening, if the plane is available, I say let's do it. But otherwise, maybe we take a year off. All right, Erin says thumbs up. So, you know, it's on. I consider, Melissa, you can anticipate a request for permission email from me. We're doing it.
If you need a backup plane, Copperhead has 2.
I mean, do you have, yes, I do need them. I mean, I'll take all the planes, send them here. I got no money, just so you know.
All right, that's great. Thank you. I see just another question in the, or a comment maybe in the chat about the eDNA project. And that's, Rich was saying that similar work's being completed in Michigan. Might be good to check and see if they detect potential hibernaculus up there. James, did you have a response to that?
Yeah, we're aware of some of that work, and I think they've been fortunate to document some near that cave. I think they're sampling water coming out of the cave, maybe, or mine. We're also aware of a study. I think they just published the findings where they documented big brown bats from water samples collected in a stream out west. So we're aware of both of those studies.
All right, thanks, James. There's a question in the Q&A. Is DNR planning to do some sort of a summary report on the acoustic survey results over the past 10 years?
Yeah, I can take that one, Melissa. We are. Sarah actually pulled an initial report together before she left last year. So we do have that posted on our website for 2020. So you can find that on the Bat Acoustic website, but we will be updating that information to include the information that Copperhead has presented.
Thank you. And I see one more question in the Q&A. This is for you, Gregg. Are the acoustic surveys still going on, or is that particular project concluded, I guess, for Gregg or Erin or Eileen? And also, we used to volunteer with the acoustic route that went around Cedar Creek State Park for several years, but then heard that the route was dropped. My office is happy to help in the Warren County area. This is from Melissa Profit. So is that going to be ongoing?
Great. Thanks, Melissa, for volunteering again. We definitely appreciate it. So what we're doing right now is we're in a transition phase, going from the old acoustic routes that we have done for those 10 years, and now switching over to the North American BAT methodology, which is basically, international, so that we're all kind of doing the same thing. So we're still working through the routes and the methods of that. But Eileen will be reaching out to volunteers here in the next month or two to start lining folks up, and we'll definitely keep your name in mind and reach out to our volunteers to get all that figured out soon. So thanks.
All right, thank you. I saw one more thing pop up. What about a ballpark cost for the eDNA analysis? So Nathaniel or James, if you know that.
Yeah, I can speak to that. It depends on a number of factors. For example, the type of analysis you're doing, the qPCR versus the barcoding, but typically a lab cost will be somewhere around $150 to $200 per sample.
Okay, thank you. That covers all the questions.
Thanks, Melissa. And thanks again to all of our lightning presenters. Okay, and last on our agenda, we have agency updates and announcements. We do this every year where we give some more agencies a chance to kind of just share what they have going on and anything that they think those of us in the working group need to know. So first up, we have Erin Hazelton.
Hi, everyone. Hi again, everyone. So really no big updates from Division of Wildlife at this time. you've heard from, all day about what we've been up to in the past year and kind of what we're looking forward to do in the coming year for research. We've had the little brown and the tricolored bat listed as endangered for the past 2 years. So, working on understanding their habitat and their biology a little bit more so we can offer better recommendations. So about our recommendations this year for mist netting, for bat exclusion, that type of thing. I don't really foresee any big changes from last year. I don't see that COVID has changed our recommendations for mist netting, so all of that will still be allowed as planned. Bat exclusion, for those of you who may be involved in that, I think we are going to give some clarification on the timing of when an exclusion device can be put on to a structure. I think there's a little bit of confusion this past year on how late in the season you could do it and still have an effective exclusion. So we'll be doing that, but for the most part, I don't think there should be any big changes. And acoustic routes, again, we're going to be switching over to the NABAT model, which we really kind of kicked off last year. So we'll be working on that, hopefully getting a few more routes out there under that protocol. But again, it's just kind of a slow process, but Eileen will be working on that this year. So I guess without further ado, the big news I have is that we do have a new biologist on staff. Eileen Weisa joined us about 3 weeks ago, and she is currently, she's a PhD student finishing up her PhD at Ohio University in ecology. And she's going to be helping us with our bat conservation efforts, as well as herps and some pollinator work. So Eileen, if you just want to say hi to everyone or say a few words if you want to, that'd be great. But I just wanted you all to be introduced to Eileen, and she'll be reaching out to you, I'm sure, as the months go on and we transition a few things onto her plate.
Yeah, hello everyone. Some of you, mostly the OU crew, probably at least knows me in passing. I'm a bit of an elusive soul, but I'm glad to be with the group now here with the DNR. I'm sure that Joe was over there laughing maniacally because he always told me that bats were best. And here I am now working with bats instead of carnivores, which is what I was trained in. So that's one victory for Joe. But I'm excited to be working with bats. Bats are awesome. It sounds like I have pretty big shoes to fill here, but I'm really excited to be a part of it and to keep going with some of these projects and to build some new stuff too.
Thanks so much, Eileen. And I think that's pretty much all that I had. I don't know if there are time for questions or if we'll just do that at the end, Marnie, but I'll turn it back over to you or over to Angie.
No, we can certainly, if anybody has any questions for Erin and Eileen, welcome Eileen. We definitely have time for some questions. Okay,
I don't see any questions right now, but I was going to point to the fact that Sarah put in the chat a link to the meeting evaluation. So just so give you guys a heads up that it's in there.
Don't leave yet, but if you do have to leave, take the evaluation before you go. Okay, if you have a question for Erin, just pop it in the chat box or the Q&A. We're going to move on to Angie Boer. She's with U.S. Fish and Wildlife Service. Angie, thanks so much for coming today and tell us a little bit about what's going on from your end.
Okay, I'm going to try to share my screen. See if this works.
You should be able to. Yep.
Okay, you can see my presentation.
Yep, just put it in slideshow mode and we should be good to go.
Slideshow. Okay, there we go. Yay. So yeah, I'm Angela Boyer. I'm with the U.S. Fish and Wildlife Service and our Ohio Ecological Services Field Office. I have just a few updates today. And I'll start off with, how do I transition that next slide?
Yeah, we're still seeing just the PowerPoint, like it's not in presentation mode.
Okay.
I mean, it's not a huge deal. We can deal with it if you can't figure it out. It's just that you can see your notes on the bottom.
Yeah, they're not even my notes in most cases. I stole this presentation.
Okay, if you don't mind, we can, it's fine. We can still see your slides.
Okay, so can you see my next slide? I'm still seeing
yes.
the other. So this one, so I want to let folks know that the Indiana bat and Northern Long-eared bat updated survey guidance. Okay. Maybe this will help. Should be coming out hopefully by the end of February. And so be looking for that. And it is going to have, you know, specific survey guidance for Northern Long-eared bat. Next slide. Yeah, and if you can see the notes, ignore them because again, they're really not my notes. And then just an update on the Northern Long-eared bat litigation. I've heard rumors that people say, oh, it's the 4D rule is going away, or it's not listed as threatened, or it's endangered. Well, what's going on with that is the court ordered us to reevaluate our listing evaluation. So it did not vacate the current listing as threatened with the 4D rule. But we are on the hook to make a new determination, a new evaluation later this year because of that court order.
Hey, Angie, we are still on the Indiana bat and Northern Long-eared bat slide. So we're seeing the PowerPoint, you know, what it looks like before you put it into slideshow. Now, okay, now we're, yeah, if you just switch through that way, then we'll be fine.
Okay.
So now we're
Sorry about
seeing
that.
the Northern. Yep, no, that's fine.
So in addition to the Northern Long-eared bat, we're also evaluating the little brown bat and the tricolored bat for to make a decision on whether they should be listed or not. And the little brown bat review is discretionary. That means we were not petitioned to do it, but we have it on our listing work plan that it should be done. And we also, we were petitioned to list the tricolored bat. So both of those species are undergoing that listing evaluation. And right now they're scheduled to have a determination of whether they should be listed or not by sometime in this fiscal year. So later on, sometime in the fall, early winter. Let's see. Okay. So just to recap that, we've got species status assessment going on for little brown bat and tricolored bats. And we're doing an SSA as well for the Northern Long-eared bat to help us make that listing decision that the court mandated us to do. And I don't know if you can see this slide because I'm not seeing it when I look at the but the leads, in case you can't see this, I'll just read it. The leads for the individual species, for little brown bat, that's Rich Gaboy. Northern Long-eared bat, that's still Jill Utrip. And the tricolored bat lead biologist is Melinda Turner. I don't know. I'm sorry. I don't know why I can't get this.
We can see that. So we were able to see that last slide with the leads, and we can see the whatever the next, yeah, the SSA framework. We can see
okay, good. And these days the SSA term is getting thrown around a lot. So I just wanted to just briefly let folks know what that is. I mean, we've been doing status assessments on species for years, but now we're doing it under the specific species status assessment framework. And it's what it is. It's just an analytical approach for assessing a species biological condition. And we do it in this way so that we can describe the viability of a species in a way that really supports our Endangered Species Act decisions. We do SSAs a lot for listing, but we also do them for the purposes of recovery planning. They can be used to inform habitat conservation plans, delisting, all those kinds of things. So it's not just a tool we use for listing, but it's really a tool that we have to help us with all of our Endangered Species Act decisions. And what we do with a species status assessment is we summarize the current status of a species, and then we evaluate into the future under different scenarios, such as a best case or a worst case scenario, to try to show what the condition of the species may be into the future. And in these species status assessments, they provide the best. It's like a one-stop shop for providing the best available science, and then it helps our managers apply our Endangered Species Act regulatory framework to the science. And the potential outcomes of the listing evaluations for the 3 bat species is there for each species. We could determine that either listing is warranted as endangered, a listing is warranted as threatened, or that listing is not warranted at all. And that just depends on what the current and potential future status of the species could be. So if a species does not meet the definition of an endangered species or a threatened species, we will not propose it for listing. So as I mentioned, the decisions for all 3 of the bats are planned for later this year. And we will make those reports available publicly when our determinations are made. So, and I also want to thank anyone who may have submitted data. Probably a bunch of you out there through your survey reports and other work that you've done have really helped provide data for those reports. And with that, I think that's all I have, but I certainly will answer any questions anyone may have.
Thanks, Angie. Any questions?
Looks like everybody did a great job of providing information. We don't have any additional questions.
Awesome. Great. Well, thanks again, Angie. It's always good to hear where you all are at with those listings. I know I get questions when I do programs, so very helpful. Okay. Last, we have Katrina with USDA Forest Service. She's going to give us a little bit of an update of what's going on in her neck of the woods.
Hey, everybody. I am the lucky one that gets to close out the meeting. I'm going to share my screen. Let's see which one. The way this looks. Okay. Oh, got to push share. There we go. All right. Can you guys see my presentation? See if I can get it to launch. Launch. There we go.
We're seeing, like we were with Angie, we're seeing your, there it is. Okay, just a little bit of delay. We see it.
Yep,
Good to go.
Definitely a lag. I've been noticing that today. All righty. So just a brief, I haven't given an agency update the last few years. I didn't really have a whole lot to say, but I got a couple of things this year that I thought I would send your way. First off, for those of you that may or may not know where the Wayne National Forest is, it's in Southeast Ohio in 12 counties. Now, this is just showing the proclamation boundary. It's not, we don't have solid ownership or anything like that. It's very much of a patchwork ownership with private lands interspersed, also some state lands and even some industrial forest. So I'm just going to quick talk about a bat conservation strategy that the Forest Service is working on. I just got done with the Lawrence County Hibernaculum Census Biennial on Friday, so I've got those results, and an ABAT acoustic monitoring. So the Forest Service has embarked for the Eastern and Southern region. So that's region 9 is Eastern and Southern is region 8. So pretty much encompasses everything east of the Mississippi, plus a little on the other side. So the Eastern United States, we've joined up and we're working with Fish and Wildlife Service to complete a bat conservation strategy, exactly what Angie was just talking about. The northern long-eared bat, tricolored bat, and the little browns are being considered for federal listing, well, northern long-ears for potential uplisting. All affected by white nose syndrome, and then throw Indiana bats in the mix. Most of us have already been dealing with them for years, but but it's getting more interesting as more bats become listed. And the Forest Service thought that it would be useful to be more proactive, not necessarily waiting to see what the listing is for the species. And we're eventually going to try to incorporate this strategy into 34 different forest plans that are covered by 30 different national forests in the eastern United States. So we're leveraging a couple of things that the Forest Service, well, we have a mission for land management, amongst other things, multiple use agencies. So we do a lot more than just forest management. There's recreation, there's a minerals program, heritage. We've got wildlife and botany programs. We've got engineers, a whole slew of different people doing different things on the land. But we have direct control over habitat management on National Forest System lands. And then, so we think that we can find ways to proactively manage bats while also meeting the mission of Forest Service to manage our forests for in the face of climate change. So we want resilient, healthy forests that provide habitat for a whole range of species and bats among them. And our goals and outcomes, again, we're wanting to protect these 4 bats. We want to maintain and develop habitat conditions conducive to healthy bat populations. And we need to land somewhere that's legally and scientifically credible. So once it's done, the plan, so we're right in the heart, in the middle of developing this conservation strategy. So there's still a lot to work out, and you're not going to see it yet. I'm not sure when. But eventually we want to incorporate this strategy into all these 34 different national forest plans that guide forest management. And then we're, as a part of that process, working with Fish and Wildlife Service to develop a programmatic consultation so that our ongoing land management activities are covered under the ESA. And this is supposed to be a living document that can be updated as new science hits the scene, as we learn more about some of those species like tricolored bats. Some of the talks today have made it clear that we don't know all there is to know yet on some of these species that used to be very common. All of our energy went towards those listed species, and now common bats are becoming listed too. And then the strategy is hopefully going to increase consistency in the application of what we're going to call the best management practices for the Eastern United States. And then, so it's consistency in how national forest managers do their work, consistency in how we interface with Fish and Wildlife Service. And if this can be used as a model, we would be thrilled, you know, if we could use it, if it has a greater use than just for Forest Service. And moving on, then I want to give you the results of the Indiana bat. So the biennial census, always done in the midwinter. This is it's an abandoned limestone mine in Lawrence County. It's on the Ironton Ranger District of the Wayne. It's been gated since 2001. I think it was actually discovered in 1998 that bats were using it. And even prior to my time working on the Wayne, I actually volunteered and did some surveys way back when. And yeah, last week, this is what it looked like. It was very, very cold. I've never seen icicles like this at the mine, so that was interesting. And I'd also like to point out that 2 years ago, so 2020 census, we actually discovered that people had bypassed the gate. It would be on the screen here in the upper right corner. They managed to dig out the dirt and squeeze in up in that corner. I would just like folks to realize that we don't have very many caves in Ohio, and we don't have any caves in Southeast Ohio. They're all abandoned mines and they're not safe to go in. So we were able to get some new angle iron welded in there, and that gap closed up, and we're hoping that people respect the fact that that gate is there for a reason. The Wayne does have a forest-wide mine closure order, making it illegal to enter mines without express administrative permission. And I'm not even allowed to go in without getting authorization at multiple levels, plus survey authorization from the State and Fish and Wildlife Service. So I'm singing to the choir here. I'm sure you guys know that stuff. But so I don't have good news to share, but I wanted to, so there's a column here that shows what we found this year. There were 4 big browns in the entire mine. And just for comparison, over the years, like I said, we've been surveying this since 1999, but at different years we were able to access different parts of the mine, and we've been able to expand a little further over time. And so the numbers fluctuated a lot because some years we didn't survey the same places, each year. So I just averaged up the 2007 to 11 because those were pretty regular years where we did the same protocol each year, just to give you an idea of what it was pre-white nose. It was a priority 3 Indiana bat hibernaculum, but we haven't had any Indianas there since 2013, I think. So it was never a huge hibernaculum. It's not like the Preble County mine. And we documented white nose here in 2011. It was the first place in Ohio to document white nose. And then we started seeing declines by 2013, and by 2014, there was a complete collapse. And so this table just shows you, again, there's the pre-white nose average that was on the previous one, and then what we've been seeing. So 2022 really isn't much different than what we've been finding in all the post-white nose years. Big browns were never super common in these counts, and they're still hanging in there. They didn't look affected at all. They handle white nose and the cold temperatures a lot better than some of the other bats. So, okay. And lastly, just wanted to let folks know that we have switched protocols, just like the Division of Wildlife is starting to do. I think it was back in 2018. Yeah, we switched from the acoustic transects that we were doing to doing NA bat acoustic monitoring. And this goes for all of Region 9, which is the eastern region of the Forest Service. Unfortunately, the Wayne is only doing 1 grit cell at this point, which includes 4 nights of stationary detector surveys, and then 1 transect that gets run twice. And then Sarah had reached out last year for another driving transect over in Marietta. She had pinpointed what was my number 2 choice. I'm ready to add more grit cells into our monitoring thing, but I haven't had permission yet. So, but I did get the driving transect going, and hopefully in the future, we'll be able to also set up those 4 stationary points. And we have a partnership with, or an agreement with Bat Conservation International to do the whole Region 9's data analysis. And basically, they regularly can detect a species, big browns, reds, hoary bats, silver-haired, evening bats even, which I think is bizarre because I've never captured, I've captured 1 evening bat 2 different years during fall swarming surveys of all things. These surveys are in July. And I've heard that evening bats may be spreading across new territory, but it is strange that they pop up quite frequently in the acoustic monitoring. So maybe Lucy's right. Maybe acoustics are picking up more diversity. And there's sometimes the tricolored bats have popped up, but just like they figured out with Division of Wildlife, there just aren't very many Myotis picked up on these routes. And it partly the design factor. Originally, these acoustics were set up to get those species like red bats and hoary bats that use open areas and are being affected by wind turbines and such. That was the original purpose, but hopefully we'll figure out more ways to detect these other species in the future. So that is all I've got. I'm going to quit sharing as soon as I figure out how. Okay.
Thanks, Katrina. Got a lot on your plate, don't you?
Oh, you know, there's always lots to do, whatever.
All right. Well, does anybody have any questions for Katrina or Angie or Erin before we close up for the day?
I don't see that any questions have come in.
Okay, great. All right, well, that is all we have for you today. Thanks everybody for joining us so much. Thank you to all of our speakers. I enjoyed every talk today, and we really appreciate your time. If you could just type a quick thank you to all of our speakers in the chat box, I'm sure they would appreciate it. And just one more reminder, oh, Sarah just posted the eval link and it's now getting lost from all the thank yous. Keep posting it, Sarah. But we do have just a very quick eval for you to take. We'd appreciate that. Tell us your thoughts, especially if you have thoughts on speakers for next year. We do a call for papers, as many of you know, but we also invite guest speakers. So please share your ideas with us. Again, we will be recording, or we have been recording this meeting, and I'm hoping to get it posted sometime next week. Sarah, Melissa, Bridget, Jeff, or Katrina, any other comments or items to mention before we say goodbye?
Yeah, I would just mention to everybody, we'll send out a note about the Bat Blitz and any potential sponsorship. And just if you want to get engaged and help us out with the planning, please reach out and respond to that as well. And thank you all for attending. This is great.
Great, wonderful. So I hope you all have a good rest of the year and hoping we can see each other in person sooner rather than later. Thanks everyone and stay safe. Bye.
And I'd like to throw out a big, huge thank you to Marnie for being our moderator as per usual, but a lot of work goes in on her end behind the scenes. And thank you, Marnie.
Happy to do it.
Thanks, Marnie. Bye, everyone.